Dear students, Assalamu alaikum. I am Professor Abuhana Mustafa Kamal, and today I will start the introduction of acid-base disorder. In my last video, I discussed regarding anion gap, base access, and osmolar gap. So, let's go. Before starting acid-base disorder, first we will have to understand about our body's internal environment, that is milieu interior. The internal environment of our body is maintained by the three mechanisms. Number one, water balance. Number two, electrolyte balance. And number three, acid-base balance. The acid produced in our body. In our body, like in the stomach, all of we know that hydrochloric acid is produced. There are many more acids in our produced during the metabolic process. They can be grossly categorized into two types. Number one is volatile acid. Number two is non-volatile or fixed acid. Volatile acid that we can expel out from our body, that is the carbon dioxide. Why carbon dioxide we say acid? Because if we bind carbon dioxide with one molecule of water, we will get carbonic acid. The non-volatile fixed acids are hydrochloric acid, already we know, sulfuric acid, phosphoric acid, lactic acid, and keto acids. Among them, sulfuric acid is the 50% of total non-volatile acid. From the name we know, volatile acid can excrete through the lungs, but the non-volatile acid cannot convert into gaseous form and must be excreted through urine. On the other hand, the metabolic bases, the base formed by metabolic process, are completely non-volatile and always excreted through urine. All of we know the normal blood pH. Normal blood pH range is 6.8 to 7.8. This is the highest range, but normally we say it is in between 7.35 to 7.45. So normal blood pH is 7.35 to 45, but 6.8 to 7.8 is compatible for life, beyond which life cannot be exist. Before starting acid-base balance and acid-base disorder, we must have to recapitulate regarding the body buffer. In the blood, the common buffers are bicarbonate buffer, hemoglobin buffer, phosphate buffer, and protein buffer. In the RBC, main buffers are hemoglobin, phosphate, and bicarbonate. In plasma, bicarbonate is again the main buffer, and all of we know bicarbonate is the most potent and universal buffer, because bicarbonate is present in mostly every compartment of body fluid, as well as its concentration is very high. Okay. Then in the renal system, the main buffer is ammonia buffer. Ammonia is the unique buffer for kidney. Then phosphate and bicarbonate. For a country, all of we know that, like Bangladesh, there are three phases of defense. If any crime occurs in any part, the first line defense will be our police force. They'll come within few minutes to hours. Unfortunately, if the crime level is more, intensity is more, then second line defense our rep comes. For their coming hours needed. And lastly, if the situation is out of control near, then our army will appear. In our body defense mechanism, just like this, 3 phases body defense mechanism is present against acid-base disorder. The first line defense is body fluid buffer system, protein buffer, hemoglobin buffer. The body fluid buffer we have already named. They come within second and minute level acid-base disorder they correct. Then second line defense system is our respiratory system. It acts within minute and continue up to hours. Moderate level of acid-base disorder it can deal. But if the acid-base disorder is profuse, then our third line defense mechanism, renal system, it appear. For starting its work, it needs hours and complete within days. So, first our body fluid buffer system, second respiratory buffer system, and third is renal system. Now, how respiratory system acts? All of you know that carbon dioxide is a volatile acid and it is controlled by our respiratory system. So, in the body, if acidosis occurs, what will happen? Respiratory rate will be increased, more and more carbon dioxide will be expelled. Ultimately from the body, acid is eliminating and the acidosis condition will be neutralized. On the other hand, if body developed alkalosis, then respiratory system will retain carbon dioxide. Respiratory rate will be low. So more and more carbon dioxide will be retained in the body. More acid will be retained in the body and alkalosis condition will be neutralized. On the other hand, basic mechanism of acid-base disorder control by the kidney is number 1, bicarbonate reabsorption from the filtrate, bicarbonated regeneration in the renal cell. Number 3, hydrogen ion secretion in the filtrate. By these 3 mechanisms, it controls acid-base disorder. So let's check about the bicarbonate buffer, how it works in kidney. In the proximal tubule, if I can show you with, yes, look, here the salt, sodium bicarbonate appear in the renal tubular lumen with the filtrate. Sodium bicarbonate dissociates into sodium and bicarbonate. Bicarbonate bind with one hydrogen that is secreted from the renal tubular cell and form carbonic acid. Carbonic acid dissociates into water and carbon dioxide. Carbon dioxide diffuses back into the renal tubular cell and water passes through urine. Carbon dioxide and water again combine to form carbonic acid. It dissociates into hydrogen and bicarbonate. Hydrogen goes back to the lumen. On the other hand, from sodium bicarbonate, sodium is entered into the tubular cell. Sodium and bicarbonate both goes back to the blood. So from the whole scenario, what we have get? Actually, if we follow the scenario, we will find that in the renal tubule, one bicarbonate came, and by several mechanisms, this bicarbonate goes back to the blood. This is the what I mentioned in my previous slide, bicarbonate reabsorption by the kidney. Here in the tubular lumen, one bicarbonate appears, and by several mechanisms it goes back to the blood. Now let's discuss about the unique buffer system of kidney, that is the ammonia buffer. This ammonia buffer, you'll have to keep in mind that bicarbonate, phosphate, etc. buffer work in mild acidic disorder, but ammonia buffer, for its activation, the acidic condition will have to high level. So how it occurs? Again, let's show. Here the salt sodium chloride appears into the glomerular filtrate. Sodium chloride dissociates into sodium and chloride. By the previous same way, sodium is reabsorbed in exchange of hydrogen. Now what happens? Now a peculiar thing happens. In the extreme acidic condition in the kidney, glutamine, this glutamine breaks down into glutamate plus ammonia. All of you know that glutamine is an amino acid that contains two amino groups. When it removes one amino group, this glutamine converts into another amino acid, glutamate. So this free ammonia enters into the lumen and bind with previously formed hydrogen, converts into ammonium, and previously from the filtrate it will get chloride, will form ammonium chloride that will passes through urine. In the renal tubular cell, you will find that here water and carbon dioxide binds to form carbonic acid. You can say from where this carbon dioxide comes. Renal tubular cell is a cell. Several metabolic pathway occurs here, and from many metabolic pathway we will get carbon dioxide. A common metabolic pathway is TCA cycle. So this carbonic acid dissociates into hydrogen and bicarbonate, and by the same way sodium and bicarbonate enters into the blood. So from here, what we have come to know? Basic things here from the lumen, no bicarbonate reabsorption occurs. Look here, there is no bicarbonate. So we will not say any bicarbonate reabsorption occurs. So from where these bicarbonate appear? Yes, students, this is the pathway where bicarbonate regeneration occurs, not the reabsorption. Here, bicarbonate regeneration, water and carbon dioxide combine to form carbonic acid, dissociates into hydrogen and bicarbonate. So here what happens? Regeneration of bicarbonate, and again look, here the secretion of hydrogen ion. This hydrogen ion passes through urine. So in my previous one slide, I mentioned you how kidney regulate acid-base disorder. Increased bicarbonate reabsorption, increased bicarbonate regeneration, increased hydrogen ion secretion. Now you can understand that ammonia buffer system causes two from these three mechanisms. One is bicarbonate regeneration, another one is hydrogen ion secretion. And bicarbonate buffer system causes bicarbonate reabsorption. Understanding these mechanisms are very important to keep in mind all these renal buffer systems. Now our last buffer in kidney that we will discuss is phosphate buffer system. What happens here? Disodium monohydrogen phosphate appear in the glomerular filtrate in the lumen. This disodium monohydrogen phosphate dissociates into monosodium monohydrogen phosphate and sodium. Same way, sodium will enter into the tubular cell in exchange of hydrogen. Hydrogen will bind with monosodium monohydrogen phosphate to form monosodium dihydrogen phosphate. Look, student, previously it was disodium monohydrogen phosphate and now it is monosodium dihydrogen phosphate. That means one hydrogen attached to it, and when it will passes through urine, it means it is carrying one hydrogen ion with it. This is called the excretion or secretion of hydrogen ion. Now, as same hydrogen, water and carbon dioxide, not hydrogen, water and carbon dioxide bind to form carbonic acid, and you will have to keep in mind why repeatedly carbon dioxide and water can form carbonic acid. Again, carbonic acid dissociates into bicarbonate and hydrogen in the kidney because kidney is enriched with carbonic anhydrase enzyme. Which enzyme? Carbonic anhydrase enzyme. So now they form carbonic acid, dissociates into hydrogen and bicarbonate. That means here bicarbonate regeneration occurs. Now sodium and bicarbonate both enter into the blood. So from the phosphate buffer, like ammonia buffer, excretion of one hydrogen and regeneration of bicarbonate occurs. There are many acid-base parameters, but the basics are only 4 to 5. The most important and number one parameter is pH. And by Looking at the pH, you'll have to find out it is acidosis or alkalosis. pH less than 7.35, definitely acidosis. pH more than 7.45, definitely alkalosis. Alternate of pH is hydrogen ion concentration. Hydrogen ion concentration more means acidosis, less means alkalosis. The third parameter, here some occurs. pH will be like this, bicarbonate this, partial pressure carbon dioxide this, and this is the bicarbonate level, and this the anion gap. So normal blood pH is 7.35 to 45. Hydrogen ion concentration is 35 to 45 nanomole per liter. Partial pressure of carbon dioxide is 35 to 45 millimeter mercury. Bicarbonate level is 22 to 28 millimole per liter. Very important, these two are. Because when you'll get a case report to solve the question, there they will mention that bicarbonate level is 32, bicarbonate level is 16, like this. On the other hand, regarding partial pressure of carbon dioxide, they will mention that a patient's partial pressure of carbon dioxide is 55. Okay. And lastly, but not least, it is the anion gap. High anion gap always related with metabolic acidosis. From the Henderson-Hasselbalch equation, we know pH equal to pK plus log of salt or base by acid concentration. From that equation, we can find out the following formula of acid-base balance. As pK is constant, so we can write the equation like this, and as the carbonic acid level has a static relationship with partial pressure of carbon dioxide, that's why ultimately we can write down the equation by this way. pH proportionate to concentration of bicarbonate divided by partial pressure of carbon dioxide. You must have to remember this equation very mindfully. So, let's start the acid-base disorder. The assessment of acid-base disorder, already I have mentioned that it is carried out by measuring actually three things: either hydrogen ion concentration or pH, and bicarbonate, and partial pressure of carbon dioxide. From this four parameter, you will have to keep in mind bicarbonate always related with metabolic activity. So any primary defect in bicarbonate, it always gives the metabolic derangement. On the other hand, partial pressure carbon dioxide always related with respiratory system, and any abnormality in primary defect, primary defect of partial pressure of carbon dioxide always means respiratory derangement in acid-base disorder. So, by determining pH, we can grossly classify acid-base disorder. simple acid base disorder. There are two types of acid base disorder. One is simple acid base disorder, one is mixed pattern. We will discuss mixed pattern later. So simple acid base disorder we can divide into two: acidosis, decreased pH; alkalosis, increased pH. Acidosis can be further divided into metabolic and respiratory. Derangement in the bicarbonate, metabolic acidosis; derangement in the partial pressure carbon dioxide, respiratory acidosis. Again, derangement in the bicarbonate level, metabolic alkalosis; derangement in the partial pressure of carbon dioxide, it is the respiratory alkalosis. For giving definition in the exam board for any type of acid base disorder among these four, we can use a common formula. If it is acidosis, you will say decrease blood pH. If it is alkalosis, you will say increase blood pH. You will start with this. It is a clinical condition characterized by decreased blood pH or increased blood pH. Depends on acidosis or alkalosis. If you will find that primarily bicarbonate change and partial pressure carbon dioxide is unchanged, then you will say metabolic disorder. So what will be the definition of metabolic acidosis? It is a clinical condition characterized by decreased blood pH due to decreased bicarbonate level in blood, where partial pressure carbon dioxide will be unchanged. It is called metabolic acidosis. If it is alkalosis, you will say like this: it is a clinical condition characterized by increased blood pH due to increased serum bicarbonate level, where partial pressure carbon dioxide will be unchanged. On the other hand, if it is respiratory disorder, you will find that partial pressure carbon dioxide has been changed and bicarbonate is unchanged. So respiratory acidosis, what will be the definition? Same. It is a clinical condition characterized by decreased blood pH, where partial pressure of carbon dioxide increased and serum bicarbonate level is unchanged. Respiratory alkalosis, definition will be: it is a clinical condition characterized by increased blood pH, where partial pressure carbon dioxide is decreased and bicarbonate level is unchanged. So now we can define acidosis and alkalosis. For defining acidosis, common formula is decreased blood pH due to either decrease of blood bicarbonate. If serum bicarbonate level is decreased, it means metabolic, or rise of partial pressure of carbon dioxide. If rise of partial pressure of carbon dioxide, it is respiratory acidosis. On the other hand, for defining alkalosis, we will say increased blood pH due to increase of serum bicarbonate. If it is serum bicarbonate increase, it is metabolic alkalosis, or decrease of partial pressure of carbon dioxide occurs, it is respiratory alkalosis. So we will get four formula for all four types of simple acid base disorder. This formula is very, very, very important. Firstly, metabolic acidosis: decrease blood pH due to decrease of serum bicarbonate level, where partial pressure of carbon dioxide is unchanged. It is metabolic acidosis. Second one is respiratory acidosis. Acidosis means decreased blood pH. Respiratory means increased partial pressure of carbon dioxide and where bicarbonate level is unchanged. Now, metabolic alkalosis. Alkalosis always means increased blood pH. Metabolic means increased bicarbonate where partial pressure carbon dioxide is normal. And lastly, respiratory alkalosis. Alkalosis means increased pH. Respiratory means decreased partial pressure of carbon dioxide where bicarbonate is normal. So, students, defining acid-base disorder is very easy. Here's a formula how you will calculate a acid-base disorder. That means if the acid-base disorder fully it is given to you a case scenario, by calculation you will find out this condition is primary or already compensated. Or if a part is not given you, like if they said you'll have to find out the pH. From this equation you will able to find out the pH. So let's go. From the Henderson-Hasselbalch equation, we know pH equal to pK plus log of salt oblique base. Here the salt will be here, not there. Salt oblique base by acid. And all of we know pK of bicarbonate buffer is 6.1. And bicarbonate has a proportionate relationship with carbon dioxide. So we can say serum bicarbonate equal to 0.03 into partial pressure of carbon dioxide. So from this value, can we find out the pH of a pH of blood? Yes, we can find out. Suppose serum bicarbonate level in a case it is 16, in another case it is 20, in other case it is 30, in other case it is 40. And if we know the log value of this, we can find out the pH of the blood. How? Look here. In my first equation, we know pK is 6.1. In everywhere, pK is 6.1. First, bicarbonate given is 16, where partial pressure carbon dioxide is always 40. As in metabolic condition, metabolic acidosis, partial pressure of carbon dioxide will be unchanged. So, carbonic acid will be 1.2 log of bicarbonate by carbonic acid is like this formula. You will get in the first equation log of 13.33. We know the log value of 13.33 is 1.12 by addition of pK with this we will get the pH. This pH means if the serum bicarbonate level is 16 and partial pressure of carbon dioxide is 40, then in the blood acidic condition will be persist and the pH of blood will be 7.22. Now, what will happen if the serum bicarbonate level is 20? All other parameters are same. If we go through the equation, we will finally find that log of 16.67 is 1.2. If we combine 1.2 with 6.1, we will get 7.3. It means if the bicarbonate level is 20, still acidic condition is persist, but it is mild than the previous one. Same way, if the bicarbonate is 30, we will get the pH 7.5. It means blood has become alkaline. Okay dear, I think you have understood how to calculate pH from the bicarbonate, partial pressure carbon dioxide and carbonic acid level if they are given. Thank you very much. Hope you have enjoyed it. If you like the video, please subscribe. In my next class, I will go for the metabolic acidosis, metabolic alkalosis, respiratory acidosis, respiratory alkalosis, and finally, I will discuss regarding the mixed acid-base disorder. Thank you very much.